All right, so next we have Michael Eggleton from.
Okay. Well, thank you. I'm very glad to be here today. I appreciate the invitation from Charlie. My name is Mike Eggleton. I'm a fisheries professor here at UA Pine Bluff, and I'm joined today by one of my graduate students, Cooper Barshinger. He's going to speak to you here in a minute about one of the projects we've done here. It's actually an integral part of this thesis. I want to acknowledge all of my partners up on the screen here. These are various graduate students and research associates and another professor that have worked on these different projects over time. Here at UA Pine Bluff, we've been involved in a number of research projects the last few years that involve big-headed carp. Now we call them invasive carps, and although there's two main species, the bighead and the silver carp, it seems like in our Arkansas rivers, about 98% of the big-headed carps we collect are indeed silver carp. I won't get into too many details. I think most of us are familiar with silver carp, their history here. They've expanded throughout the Mississippi River Basin, and there are a number of impacts. They are highly planktivorous, and the fear is they compete with lots of native fishes and juveniles of most species. They have pretty broad tolerance for various environmental factors. There are declines in native fish condition that they think have been linked to the presence of lots of silver carp. There's possibly been shifts in native fish assemblages, and there are some sport and commercial fisheries declines that are suspected to be linked to silver carp establishment in some systems. The ANS Small Grants Program has funded several prior projects here at UA Pine Bluff, both directly and indirectly. The first and the fourth project I list here were funded directly by the Small Grants Program. The second and third projects were actually funded indirectly. The fact we had Small Grants Program money enabled us to leverage some other money. Cindy mentioned a little bit of that about that this morning. We've been able to leverage some other sources of funding to do some other projects. And I'm going to give you an overview of these projects here today. The first project I want to talk about was a project we did a few years ago, and it looked at invasive carps in the Lower White River. The Lower White River is over in eastern Arkansas. It's a very lowland system. The White River rises in northwest Arkansas, goes up into Missouri and back down. But in eastern Arkansas, where we worked, it's very much a lowland river. We worked mostly in the White River National Wildlife Refuge. Silver carp were very rare in that system until about 2010. There was an historical study done, another master's thesis back in 2002. It only collected 2 silver carp, and there was plenty of sampling in that study, so if they were there, we would have gotten them. We did a lot of multiple gear fish sampling, very thoroughly characterized about 15 oxbow lakes in the refuge. We did lots of multivariate statistics, and the whole study design here was emphasized to look at what we called the pre-carp fish assemblages from 2002 versus the post-carp fish assemblages that we measured in 2017. It was a very fun project. We did a lot of fish sampling in the White River, a number of different gears, got to see a lot of fish, and that's always what a fish squeezer likes to see. We did a lot of fish sampling in this White River project, and a lot of these numbers here are too many to digest, but over here on the right side, these are the total numbers, and when you look at the overall samples from 2017 and 2002, post-carp versus pre-carp, we see roughly the same number of species were collected from these lakes. The diversity, evenness, and dominance values were very similar. Unfortunately, a few of the extra species we did collect in 2017 were actually invasive carp species. We picked up a black carp and some grass carp and a few bighead carp, I believe. So that was part of our increase in diversity, were actually the invasive carps we were not really wanting to see. But nonetheless, this very gross, very coarse measurement of assemblage structure really doesn't tell us very much. Okay. When we look at assemblage structure in a more detailed manner, a much finer grain look at it, we did some multivariate ordination, non-metric multidimensional scaling, and this is on our electrofishing data. And if you've never seen this type of analysis before, each coordinate or label that you see on here is a coordinate that represents the fish assemblage at that location. And coordinates that are very close together tend to have very similar fish assemblages, and those that are far apart tend to be very different. The first few letters of each of these labels are simply the lake name, but the 02 or the 17 at the end is when the data were collected. And as you can see, the 2002 and the 2017 data sets were quite different. In the case of our electrofishing data, it appeared the pre-carp samples tended to have slightly more gizzard shad, white crappie, and to a lesser extent, yellow bass and bluegill. And then the post-carp samples from 2017, we tended to see fewer bluegill, about half as many as in 2002, but we did see some other sunfishes in slightly greater abundances, as well as bigmouth buffalo, a weed shiner, and spotted bass. So sort of a mix of species here. There were some omnivores, some insectivores, and even a predator. So there were some real differences there, and multi-response permutation group tests said that those assemblages were different. When we move on to the mini pike net data, mini pikes are a littoral zone gear. You sample them in very shallow water, and that's not typically where silver carp are found. Nonetheless, our mini pike net assemblage data were also significantly different, but they were much more similar. Between the pre-carp and the post-carp periods. They were very, very similar, even though they did test significantly different with MRPP. And these species I have listed here, which are all small-bodied littoral zone species, you tended to see more brook silverside, emerald shiner, orange-spotted sunfish, and pug-nosed minnow in the pre-carp samples, with a slight preference for weed shiner and mimic shiner during the post-carp. Now, this doesn't mean that, for instance, mimic shiner were never found in the historical data. It just means there was a slight increase in abundance during the post-carp period. When we look at gill nets, we're back to like the electrofishing data. The assemblages are very different from one another. Pre-carp samples tended to have more gizzard shad, white crappie, and to some extent yellow bass. And the post-carp samples tended to be a little more dominated with freshwater drum, small and bigmouth buffalo, and then long nose, short nose, and spotted gar. So we saw some pretty stark differences here in assemblage structure. We did notice there were 8 species not found in 2017 with the carps compared to the historical data sets pre-carp, and then 2 net new species were collected in 2017. These species that were lost and gained weren't really all that important. We don't think they were all historically rare, and we think it was probably more of a gear selectivity or possibly a small seasonal difference. The MRPP group test did indicate significant shifts in the assemblage structures between 2002 and 2017. And 2 of the 3 gears suggested very strong differences, and the differences were less with mini pikes. Now, whatever we find here, we can never unequivocally say these are carp effects. That's what we're looking for. Trying to get my frame to advance here. There it is. Okay, what we don't know about this White River system, you know, these types of structural shifts in fish assemblages, they can happen every year. We don't know that, and there's maybe not a good reason they would, but again, we can't verify that they don't. Okay, it looks like it jumped ahead on me. That's okay. We have 2 good years of data, but from a 16-year time span. So again, we can't totally write this off to the carps. We do not think carp abundance is the whole story. We think they probably interact with other factors, and you know, we hope to do further research on that. The second project I'll talk about is related to one of the later projects that you'll hear about, and we wanted to learn about silver carp population dynamics in Arkansas rivers. A lot of the silver carp work historically has been done in the upper Mississippi River Basin. It is being assessed in different places in the LMR Basin, but there isn't a lot of information quantifying growth, mortality, recruitment, and things like that, though there are people working on that. In this study, we collected a lot of carps from 5 different river systems during 2019 and 2020. Our goal was to collect at least 100 silver carp per system, and we did. We captured them, we sexed them, we measured them for length and weight. Lapilli otoliths are actually better for aging silver carps than sagitta, so we extracted those in the field, and there was a lot of aging done in the lab, double blind, read whole view, and sectioned. And I won't get into too many details about that, but the sectioned readings are what we considered to be the true age. Okay, I'm not getting my frame to advance here, so I'm going to try to move on. Our question was, we wanted to see, you know, how much these vital rates of population dynamics, how much they varied across these different river systems. And I can't go through everything today, but we did measure condition We looked at size structure, a couple of growth modeling. We also did back calculated growth and length of age. We also quantified mortality with a lot of catch curves, and we quantified recruitment variability. And I'm having trouble getting an advance here. Okay, I just went through that. I'll move on. Yeah, the 5 study rivers, well, just skipped over. Okay, I guess I've got to slow down. There's a really slow pause here. The 5 rivers we looked at were the lower Mississippi River proper itself, a couple of different locations down around the mouth of the Arkansas River. We also worked in the Arkansas River itself from the mouth all the way up to Little Rock. We worked a pretty good section of the White River from Clarendon downstream. There were some fish from Batesville, which is further upstream. We worked in a couple of locations in the Cache River, which is further north and in the White River Basin, and then a couple of locations in the St. Francis River. In the end, we collected 552 carp for this analysis. They were collected between June of 2019 and November 2020. Some of the specimens were provided by third parties. We did have some arrangements with biologists and even commercial fishermen that did provide silver carp specimens for us. The aging results, I won't get into details here. Initially, they were really horrible. The 2 readers only agreed about 32% of the time. However, 81% of their disagreements were only by 1 year. And for populations that had up to 13 age classes, that was not really bad. We found out that disagreements were about equally distributed amongst young and old carps, and from the other aging work we've done with other species, that's pretty unusual. Usually it's the old fish you get the most disagreements on. These guys were tough enough that we found disagreements everywhere. But again, our readers eventually worked together and resolved a lot of these discrepancies, and there were a lot of advances made in improving our aging techniques. This is an unusual species there's not a lot of work been done in this area, so they were difficult to do in the beginning. Okay. Oh, it did let me advance back. Okay, I'll look first at mean size. The Arkansas River tends to be our largest population. They tend to also have the greatest size variation, as you can see in this length frequency. I think all 25 fish above 1,000 millimeters that we collected were from the Arkansas River. You can see great variability. The Cache River, pretty large size structure, but not quite as much variation. The lower Mississippi River and the White, moderate size structure and kind of a similar distribution. The St. Francis River, a smaller Mississippi River tributary, tended to have a smaller size structure. And you can see, again, the Arkansas River being our largest population is kind of a recurring theme. When we moved to age structure, we collected 13 age classes. We never collected any age 1 or 2. That's been pretty common in a lot of studies. We do only get a handful of age 3s. They don't seem to recruit to our sampling gear till about age 4. I skipped over that frame, but this was all done with boat-mounted electrofishing. The mean ages of these fish around 7 years, ranging from about 6.2 in the White River to 7.8 in the Arkansas, and the age 5 through 12 cohorts comprised about 90% of our catches. We fitted a lot of von Bertalanffy growth models. Now, our overall model from all 552 fish, we generated an L infinity, which is a theoretical maximum size of 916 millimeters. Our so-called growth coefficient was about negative 0.19. That's modest. And a T-sub-O is sort of an adjustment parameter, negative 0.87. Those are frequently very small negative numbers, and this one worked out about the same. So these curves, again, the Arkansas River kind of stands out. They tend to be larger, perhaps grow a little faster, and then the St. Francis tends to be one of our lower populations. The White River here seems to be stuck in the middle. When you look at the individual models, and again, I don't want to blow you away with all these numbers. I want to draw your attention to the Arkansas and the White River. Okay, I need to go back. There we go. I advanced one too further. The Arkansas River, again, our largest population. The growth coefficient here is lowest, but these are negative numbers, and so it's a greater number. But when you plug this into a von Bertalanffy model, L infinity drives your growth, and even this lower looking growth coefficient will generate bigger fish. The Arkansas River fish at length age 6 are quite a bit bigger, a couple of 100 millimeters larger than these other rivers. So it is a larger, faster growing population. The White River tends to be at the other end of that spectrum. It tends to be one of our smaller and slower growing population. So that's kind of interesting because carps are very abundant in both. In terms of mortality, the Arkansas River, again, in the white kind of stand out. 12% annual mortality from a catch curve in the Arkansas, up to 36 in the white. Overall, about 28% across all these rivers. That's kind of a nominal annual mortality estimate for a lot of species. And looking at the recruitment variation index, it gives you an index of recruitment stability. Recruitment is pretty stable in all these populations, but our greatest stability was in the Arkansas, and our least stability, but still yet very stable, was the White River. Okay, I'm advancing on here. So the conclusion from this, we tend to see our Arkansas River silver carp populations, they look healthy and viable. The recruitment appears to be pretty stable in all of these rivers. The Arkansas River consistently stood out, large sizes, low mortality, very stable recruitment. This is a bit surprising since it is a serial lock and dam system that has lots of barriers to upstream migration. You collect fish intermittently all the way up to Little Rock, but still there's a lot of hurdles to get through, and it's very surprising to see them get as far up in that system as they do. We're not finding juvenile or young carps really anywhere. We've never gotten an age 1 or 2, a handful of age 3s and 4s. We don't really start picking them up till age 5. I didn't mention that because I think that frame got skipped over. Our catch curves included only fish at age 5 to 12. That was 90% of our catch, and the cohorts younger than age 5 appear to be underrepresented. However, we see no reason at the present time to not think all 5 of these rivers will continue to have healthy populations. I know that's not what people want to hear. How does this river, the Arkansas rivers, not the river, but the rivers that we worked in, the 5, how do they compare to other populations that have been more thoroughly studied? Well, the upper Mississippi River tends to have greater sizes and L infinities, but the recruitment is stable, like we see down here. The Illinois and the Wabash rivers in the Midwest tend to be smaller, lots more of them, but they're smaller, lower L infinities. They too have very stable recruitment. Work done in the Missouri River up in South Dakota, they tend to be smaller and they grow less, and the recruitment does tend to be a little less stable than what we see down here. And then the Tennessee and Cumberland rivers over in Tennessee and Kentucky, they too do tend to be a little bigger with larger L infinity values, but their recruitment also looks to be a little bit less stable than what we see in Arkansas rivers. So the Arkansas rivers that we worked in tend to be kind of in the middle of the pack, which is something we needed to know. There was very little information before this, and now we sort of know where we measure up, and we're going to keep wrestling with that. Okay, the next study I want to talk about. Okay, it's already jumped ahead. Let's see if I can go back. Yes, this is a project that we just got started. Now, this is a project indirectly funded by the Small Grants Program. It's a study design similar to what we used in the White River, but we had access to an historical data set from 7 lower Mississippi River secondary channel locations spanning from Kentucky and Missouri down to Mississippi and Louisiana. I worked on that project as a graduate student back in the 1990s at Mississippi State. There were no silver carps collected in that study. Very few big head carps, some grass carp, that was really about it. Those same areas are overrun with silver carp nowadays. So we were able to put some grant money together and leverage some other sources, and we made a project, and we are looking at those historical sampling locations yet again from Kentucky down to Louisiana. And we are going to have the same sort of study design we did in the White River. We're going to emphasize the pre-carp and post-carp fish assemblages from these habitats, pre-carp being from the 1990s. Silver carp were out there, but they weren't very abundant. And then we just started this project last summer and fall, and we're going to continue it for a couple of more years. I just have, these data are very, very preliminary, but we did some more multivariate ordination, some non-metric multidimensional scaling. You look over here on the left, that's 3 years of data back from the 1990s. And again, these are sampling locations with the year at the end here, and you can see a lot of 90s, 95, 96, 97. And then the 2021 samples, now there's not very many of them, but they tend to be different, at least in multivariate space. Now, these are a very imbalanced data set. The red, not very much data. This is a lot of data. So it's not a perfect analysis right now. We've got a couple of more years to work on this. As far as what species are driving those differences, Again, we only have preliminary information, but the 2021 sampling we just completed last year, silver carp tend to be a pretty prominent species. They're about half the fish biomass in most locations. There does seem to be a weak association with blue sucker, skipjack herring, and long-nosed gar, as well as smallmouth buffalo and short-nosed gar. That doesn't mean they're not found when there's no silver carp. It just means that when you find higher numbers of silver carp, you tend to see a little more of these species as well. As far as what we found in the historical samples, in the absence of silver carp, it was really just the common species: common carp, channel catfish, river carp sucker, freshwater drum, and goldeye. None of these species have disappeared. They just tended to be a little bit more abundant, it seems, in the historical samples. This is just a first cut analysis on these data. We have a lot more to look into. So okay, I'm going to shift gears now, and I'm going to turn the talk over to Cooper Barshinger. He is my graduate student. He's going to talk about the fourth and final project that was funded directly by the Small Grants Program, and this is actually a big part of his thesis, and he's working on it right now. So, Cooper, I'm going to let you move in here.
Okay, folks, so our fourth project looked at figuring out the natal origin of silver carp, and within the lower Mississippi River, we have multiple tributaries, as we've talked about in some of the other studies. And at this time, we have no knowledge of how silver carp's life history may be using these tributaries. It's been known that tributary systems in silver carp can be spawning areas during flooding. And to look at this within the lower Mississippi River, we're looking at using microchemistry. So otolith microchemistry is used by knowing that otoliths are inert following annual accumulation of the calcium carbonate matrix. And as these layers are added on, most people will use these for aging fish, but you can also use them to figure out where the fish have been. And this is possible due to the trace elements that are imprinted on the otoliths from the water that the fish are in. And these trace elements come into the water depending on what geology that the water is flowing through. So oftentimes for silver carp otolith microchemistry, we use the lapilli otolith as we did for the aging. So our second project is corresponded with this fourth project very heavily. And we see that the lapilli otolith has a greater space in between the molecules seen in the top right picture, and that has an aragonite structure compared to the sagittal otolith. Overall, when we're looking at microchemistry, we're looking at the elements of strontium, barium, and magnesium. And studies like this have been done in the upper Mississippi and the Illinois, such as Norman and Whitledge's study in 2015. So to prepare our otoliths, we used a similar way as we did for sectioning the otoliths, and that was using isomet low-speed precision saw to get the otoliths sectioned as seen in the bottom right picture, and then we polished them and sanded them to get them ready to send to the lab to be analyzed. And with that, we used a ICP-MS to analyze these otoliths, an inductive coupled plasma mass spectrometer, which is shown in the bottom right picture. The laser ablated a transect across the otolith core and measured the strontium, barium, and calcium. And then we also got a one-spot ablation measured at the core as well. We also took water samples during 2019 and 2020 that were used or analyzed using ICP-MS as well for strontium, barium, and calcium. Our water results are shown here. The among river differences for strontium calcium were found to be greatest between the Arkansas and the White, with a ANOVA of significant differences found between the all of the systems were found to be significantly different, and the three that we're really looking at are the Arkansas, the White, and the Mississippi, which are our largest tributaries. And the Mississippi kind of fell in with those other river systems in between the Arkansas and the White. And when we ran our Tukey HSD, we found that there were 5 different groups represented by those letters below the boxes. For our barium study, or ANOVA, we've again found a significant difference. The Arkansas and the White were not found to be as different, but there was still a difference when you looked at the Mississippi and the White compared to the Arkansas. The greatest difference was between the White and the Cache, but when we looked at all of these for the otoliths, we found that really the biggest differences that we were going to see were between our Arkansas, White, and Mississippi River. So that's what we ran with for the otolith microchemistry. And when we ran the strontium, we found that all 3 were significantly different. Our Tukey HSD test also showed this. When we went into the barium, we found that it was not quite as different, but the Mississippi, just like the water sample, was different from the Arkansas. And that played a key role in making our model prediction. When we look at our classification tree diagram here, we have the Arkansas starting out there at the top. The percentages below represent the Arkansas, Mississippi, and the White. The Arkansas and the Mississippi had the same number of fish for the samples that we used for microchemistry. The Mississippi didn't have quite as much, and that's why that number is low. As you go down, any value that was below 743 strontium got knocked over into our White branch. Anything that was above that went over to the Arkansas branch, which was then split between the barium if it was greater or less than 39. It went down into the Mississippi or the Arkansas. And to make this model, we used the outer layer. We were able to check it for its accuracy by knowing where the fish was collected. And we got an 80.2% accuracy. And we then put our spot ablation, which was the core, into this model to get our natal origin. And we found that a majority of our fish are coming from the Mississippi and the White for their predicted natal origin. And the Arkansas seems to be not as much of a factor in having spawning or natal origin for our silver carp here within the lower Mississippi River. And as we go into our discussion here, we see that if you add up the White and the Arkansas, almost 80% of our fish have a natal origin in those 2 tributaries. At this time, we have little information other than this microchemistry study to go off of for making predictions for management on where the fish may be spawning. But at this time, it does not look like the Arkansas River is going to be playing a big role in controlling the populations if we would be focusing on eliminating these fish. The smaller river systems may be a possibility to include in further studies if we're going to increase our water samples, but at this time it's really hard to distinguish those from the larger systems, and when we do put them into the model, our accuracy goes down very far, almost to the extent that you might as well just flip a coin at 50% accuracy. So with that, I'd like to thank our people that helped out and acknowledge the University of Arkansas at Pine Bluff, the U.S. Fish and Wildlife Service, the small grants program here, Angie Rogers with the U.S. Fish and Wildlife Service, the LMRCC, James Ballard with the Gulf States Marine Fisheries Commission, Jimmy Barnett with the Arkansas Game and Fish Commission, and Mr. Chad Washington with the Mississippi Department of Wildlife, Fisheries, and Parks.
Is there any questions of the group? I had a quick question. I don't know if it's quick. I saw there's a different size structure. I think a lot of what you just presented answered some of that. Between the rivers, there's a size structure differences, and you kind of pointed that out.
Yes, sir.
Is there any specific reasons that you could point out to explain those differences in size structure? Besides what, I guess I just realized that well, I didn't realize, the spawning doesn't seem to be happening in the Arkansas River, so that might explain a lot of it.
Yeah, the microchemistry work, Cooper is still continuing to refine that. He's actually also looking at the strontium calcium and barium calcium ratios, and when you use those models, those measures in his predictive models, it actually de-emphasizes the Arkansas River even more. and emphasizes the Mississippi and the white. Principally, though, the Mississippi is where the spawning is probably taking place. However, back to your original question, the Arkansas River is a bit of an oddity. It does not look like a system that should be, it's very hard. It's lots of locks and dams, It doesn't look like it. The silver carp could do well there, but we're really puzzled by the wide range of ages and sizes that you see in that system. It just doesn't look like, given the locks and dams, it does not look like a place that silver carp would thrive, but they seem to do well there. The wide variety of sizes and ages, the high growth, it's just really vexing, a bit perplexing why we're seeing that. Typically very highly regulated systems, there is a theory out there that they're very conducive to invasion and for wide expansion of invasive species, but the Arkansas River is just really hard to imagine that, given the number of barriers that they have to get around. But so we don't really have a firm idea, but it stands out not in so many different ways. We're continuing to look at it.
Has there been any telemetry work to kind of show some patterns with that might explain it?
Yes, we're just getting that started. There is another project here at UAPB being funded from another funding source, Steve Lockman, and his students are tackling that right now. They've tagged, started last fall, putting tags in silver carp. They are tracking them. They have, they track them in boats, and then they have antennas mounted permanently affixed to bridge overpasses and things like that. They're monitoring. They're not seeing a lot of fish moving back and forth between the White and the Arkansas River through the Post Canal. They're very interested in seeing if that occurs, but at least since last fall, it does not seem to occur. Now, maybe their spring and summer movement patterns will be totally different, but their fall and winter patterns seem to be kind of limited. There's White River fish and there's Arkansas River fish, and they're kind of staying put. But yes, we have a study that's hoping to look at that, very thoroughly over the next couple of years.
Well, thanks for sharing. I appreciate it.